Okay, you can still see the screen, right? You're looking at a blue screen?
Yeah, blank blue screen now.
Okay, so what we want to start talking about now is the way a cell responds to challenges. So it becomes important, like I think I mentioned at the beginning of today, that like all things in pathology, you need to have a good handle on normal before you can appreciate what can go wrong and also predict what might happen when things do go wrong. If you don't have any idea how the muscular system works, you're not going to know what could happen with pathology. If you don't know anything about a cell, you're not going to know what can go wrong. So we know that a cell generally has a nucleus, unless we're talking about a red blood cell, and that within that nucleus we have the genetic material for the human in our example. Every cell that has a nucleus has genetic material. The genetic material is DNA. Along the chromosome, the DNA is arranged in little clusters of genes and with other DNA in between those genes. And that's going to give not only cell behavior, but overall organism behavior. It's the genes within these cells that give us features like blue eyes and brown eyes and your height and your personality and your ability to resist certain types of environmental stressors that might cause cancer in some, but not in others. All of this sort of organism presentation and behavior is genetically driven. Now, can you influence it with environment? Absolutely, you can. Can you influence the genes to either be turned on or turned off? Yes, you can. We're learning more and more about how we can do that sort of thing. But that's coming from within the nucleus of that cell. Now, the purpose of the genes at the cell level is to signal the behavior of that cell. So the genes are going to create a messenger called RNA. The DNA is creating some messenger RNA within the nucleus of the cell. The messenger RNA is going to leave that cell and it's going to signal cell behavior. Most of the time, at least part of what the cell is going to do is create new proteins. We've got enzymes, we've got hormones, we've got neurotransmitters, we've got other types of cell signal molecules that are going to be produced in one group of cells to influence another group of cells. That's going to be generated in the messaging that goes on between the DNA and the RNA. When the RNA is released from outside of the nucleus, it's going to signal ribosomes and endoplasmic reticulum to create new proteins. It can't do that unless it's often got enough energy. Energy we get from molecules that will release energy when we break them down. The most common molecule that we utilize the energy from is a molecule called ATP. Now, if you make the mistake and call ATP energy, you're going to be wrong, right? ATP itself is not energy. When we break ATP down to ADP, we get a phosphate group and we get energy, and along with that energy, we create some heat. So that's one of the mechanisms we utilize to modify or influence the homeostasis of body temperature. The more ATP we break down, the more we're going to raise the body temperature. The more metabolic activity really means more ATP breakdown, means we're going to not only get the energy, but the heat that comes with it. Now, everybody okay with that? So what it really means now is that you need to review the cell, and particularly with emphasis on the plasma membrane and understanding that the plasma membrane has integral proteins, it has transmembranous proteins, it has structural proteins that create shape and mobility and identification and mobility of the cell as a whole, but also its transport through the cell membrane. There are receptors and markers on the cell membrane that give us identification, but also match with signal molecules from other parts of the body that influence the cell's behavior. So if none of that sounds familiar, or you're kind of going, what the heck is going on here? You got to spend some time and review it. It's important that you understand that sort of thing. So a cell is a living organism, and as a living organism, it has adaptability. It has the ability to respond to its environmental signals or environmental factors around it. So living cells can respond to a stressor in a number of different ways. One of the ways that living cells respond to a stress is that they can outlast the stressor. So, for example, if a lack of oxygen to the body was the stressor, let's say you decide to go for a swim, you go underwater. Now you don't have easy exposure to oxygen from the atmosphere. There is some oxygen in water, but not the kind of oxygen that we can get through breathing. So we hold our breath. That represents a low oxygen environment. Some cells have the ability to store oxygen in them. Skeletal muscle cells store oxygen. Cardiac muscle cells store oxygen. Neurons do not. So a neuron is going to be less able to outlast the stressor, low oxygen, than a muscle cell will. So one way that cells respond to stressor is they have mechanisms that allow them to outlast the stressor. You can dive underwater, swim for a little while, come up and take a breath, and you're fine. If you're underwater too long, the first cells to be affected are going to be neurons. Brain damage from being underwater too long. That's the concern in someone that's underwater and been underwater for too long. Now, another mechanism that cells have is their ability to adapt. Sometimes a cell will respond to a stressor by changing, by adapting. So it can change its function or its structure to respond to a stressor. The easiest visual of this type of adaptability is what happens to parts of our skin when regularly exposed to friction. So let's say you are a construction worker and you're constantly working with rough abrasive materials, whether it's lumber or concrete, or you're in the dirt and you're constantly exposing your hands to friction and hard, rough surfaces. Eventually what's going to happen is you're going to grow or develop calluses. Calluses represent the epidermis of your skin adapting to that chronic rough stress. Added layers in the epidermis that will toughen and resist that stress. Now, both of these mechanisms that cells utilize are reversible. If that hard labor worker takes on a job in an office, eventually those calluses are going to be sloughed off with the outer layers of the epidermis, and you're going to be left with softer, thinner skin. No stress, no need for the callus. Okay, am I making sense so far? Are we understanding this? Somebody turn on their mic and say yes or no.
Yes.
Okay. So there's lots of other examples. We'll use some of those examples when we get into some specific systems. Now, the third way that a cell's going to respond to a stressor is that it's going to die. And cells that die cannot come back to life. So death, obviously, I hope it's obvious, is going to be an irreversible response to a stressor. This is a scenario where it's unable to overcome the stressor. It can't outlast it, and it can't adapt to get away from it. So what do we look for when a cell is being challenged? What sorts of things is a cell going to demonstrate that might indicate that it's in trouble? So we're looking at indicators. So early indicators before it becomes irreversible that indicate a cell is not successful at dealing with a stressor. So the first thing to be aware of is that generally a cell is going to undergo something called hydropic swelling. In other words, if a cell normally has a shape that looks something like that, and we look under a microscope and we see that it's quite enlarged, and where we had some organelles, that were sort of that close together, with the hydropic swelling, the organelles are now going to be further apart. So hydropic swelling just refers to the fact that the intracellular fluid volume is increasing, and eventually that's going to lead to deterioration of both function and structure in a cell. So what's the mechanism? Remember, we use that word pathophysiology to understand the mechanism of hydropic swelling. Well, what's going to happen is in any cell that exhibits hydropic swelling, it's going to start with the decrease in mitochondrial function. So what's the main responsibility of mitochondria in a typical cell? Somebody tell me what it is that mitochondria are responsible for in a typical cell. Okay, maybe I better reword this. Of the 28 people that are in class today, is there anybody who knows what the purpose of mitochondria are or is? Is there anybody who wants to try to tell me what the function of mitochondria is in a typical cell? You would have talked about this probably starting in about grade 4, grade 5, and again in middle school, and again in high school, and again.
I think the most high school response to that would be like the powerhouse. the function of the cell.
The powerhouse.
Okay, so that is the elementary school answer, that it's the powerhouse of the cell. So that's good. We'll start there. But let's face it, that's not really telling us very much. What is it that's happening inside the mitochondria that gives us the reason for us calling it the powerhouse?
So like the breakdown of like carbohydrates.
Yeah, well, breakdown, but for what purpose?
Energy.
To produce energy.
Energy.
But what's it actually producing?
The ATP.
Is it sugar?
It's,
Or
what's,
ATP, yeah.
what molecule is it actually producing?
Is it glucose?
No, you glucose, you got to eat glucose. We're not making glucose. We don't make glucose. Okay, we need to take a little break here. and we need to look at the way a cell here's our powerhouse, mitochondria. Glucose is in a blood vessel because we ate it. You talked about the digestive system. You talked about absorption of nutrients. One of those nutrients is glucose. Glucose is our favorite way to make the molecule that we're going to get energy from. But it's no good to us in the blood unless we get it into the cell. And then glucose is going to become pyruvate. And does anybody know what that process is? When glucose gets converted to pyruvate, that's called glycolysis. And we make a little bit of ATP, but not enough to be functional. So we got to get pyruvate into the mitochondria. And the way we get pyruvate into the mitochondria is we add some oxygen. And then the pyruvate is going to get converted to acetyl coenzyme A. And acetyl coenzyme A enters the Krebs cycle in the electron transport chain, and it makes lots of ATP. Lots of ATP. ATP is no good to us unless we break it down to ADP. That's a diphosphate instead of a triphosphate. So we've got a phosphate group here, and what we're looking for, energy. But that whole process generates heat as well. So ATP is not energy. Energy is what we get when we break ATP to ADP plus phosphate. And along with that energy, we're going to get heat. Now we have something that will allow us to synthesize proteins in the cell, or will allow us to endocytose, or exocytose, or actively transport into or out of the cell. Now, when you look at this in this quick little summary, is it familiar?
Yes.
Okay, to one person it's familiar. So this is what you need to review. If this is not familiar, if you're not comfortable with this level of description, you need to review it. It becomes really important that you understand these sorts of things. This is the basis that you should have learned in 101 and 201. Okay, especially 101. We did a lot of this metabolism stuff in 101, and then the actual metabolism of where glucose goes once you eat it, and what happens to it once it gets into the digestive system. That was 201. Right? You did the digestive system in 201. So both semesters you should have covered varying degrees of this kind of thing. This should be easy. So now we come back here. So I'm telling you that where hydropic swelling is going to come from starts with a decrease in mitochondrial function. If we have a decrease in mitochondrial function, what's going to happen in that cell? What is not going to be made enough of? Okay, let's go back to this one. We're going to lose time. You're going to have to do some self-study today or this week because we're spending time on this stuff, which we shouldn't have to spend. So what did I say was happening inside the mitochondria in this diagram? What do we
It's being produced.
Yeah. Sorry?
It's just the ATP is being produced.
Yeah, lots and lots of ATP gets produced in the mitochondria in the presence of oxygen. Aerobic respiration. Glycolysis was anaerobic. We didn't need oxygen, but it only makes a little bit of ATP. We need lots of ATP for cellular function. So if we go back and look at this and we say the mitochondria is no longer doing its job, decreased mitochondrial function, what is that going to mean? What are we then going to see a decrease in?
ATP production.
ATP.
Yes. It wasn't supposed to be hard. We're going to see a decrease in ATP production. Now, this is a bit scary for me. What else? What's one of the basic things that we need ATP for in virtually every cell in the body?
Cell production. Oh, you mean sodium reabsorption, potassium.
What is this thing? What is this thing I just drew right here?
Is it a cell membrane?
No, the yellow part. This whole white thing's cell membrane.
Sodium,
It's the
sodium,
ETC
potassium
chain or
pump.
something.
It's the sodium potassium pump. And the sodium potassium pump has a binding site for ATP because sodium has to constantly be pumped out of the cell. It wants to sneak back in because over here, there's a channel that allows it to sneak back in because it's in high concentration outside and low concentration inside. So if there's a pathway, it's going to flow downhill into the cell. So we need a pump, and a pump that has to go uphill is going to need energy. It needs ATP. Right? You remember that? So the goal of the sodium potassium pump is to pump sodium out of the cell and potassium into the cell. But in both cases, we're going against the concentration gradient. That means by definition, it has to be active. We need ATP. You see that? You see what I'm talking about here?
Yes.
Yeah.
You don't sound very confident about it. Okay, it's a big problem, guys. It's a big problem if this sounds brand new to you. So if we don't have enough ATP, then we're also going to have decreased sodium, potassium pump function. And what that means is sodium is going to be able to rush inside the cell because the sodium potassium pump's not working properly. And what do we know follows sodium?
Water.
Water. So we're going to get sodium rushing in, water's going to follow, and the cell is going to swell. Hydropic swelling. Now, this little pathway that we drew here doesn't help you if you don't understand what the role of the mitochondria is in the first place. And it doesn't help you if you don't understand what the role for ATP is on the sodium-potassium pump. So if you're having some trouble remembering this sort of thing, you've got to spend some time in review. Okay, so what's going to happen is the sodium concentration gets higher inside the cell. Water is going to follow the sodium inside the cell in the interest of trying to keep an equilibrium of concentration, and we're going to get swelling. If the swelling is allowed to progress, it's going to result in dysfunction. So this, we said, was an early indicator. So we're not yet at dysfunction, but when we see swollen cells, we know that this cell is failing. It is not being successful at adaptation or outlasting a stressor. It's starting to break down. So we said that a failing cell is going to exhibit hydropic swelling. And the other thing that we might see under, you need a microscope for both of these things, is what are called intracellular, so inside, intracellular accumulations. So that's just a general term saying that when you look under a microscope, you see a bunch of stuff inside the cytoplasm of that cell that wouldn't normally be there. So they could be carbohydrates or lipids or proteins that wouldn't normally expect it to be there. So you're under a microscope, you see all this sort of junk in the intracellular space, and you go, whoa, there's something going on there. And that's usually because of the swelling. It usually goes hand in hand. But you're going to, basically, it means that there's either some enzymes or some genetic abnormalities that are causing this accumulation within the cell. But it's an indicator that this cell's in trouble. So those are the two big ones that tell us that a cell's in trouble. Now, what does a cell do for adaptation? Cell adaptation. One thing a cell can do is it can shrink. Atrophy means it's going to get smaller. It may get smaller because it doesn't get enough nutrition. That's the stressor. Or it may get smaller to reduce the overall ATP demand. The bigger the cell, the more energy it needs. Smaller the cell, less energy. Maybe it can outlast the stressor based on having a smaller demand for ATP. Does that make sense? Yes, no?
Yes.
Okay. Now, what's the opposite to atrophy? Does anybody know the word?
Hypertrophy.
Yeah, hypertrophy. So sometimes what we'll see is that a cell's adaptation will be to grow bigger. And when it grows bigger, it's growing, it's sort of counterintuitive. You'd say, well, if it's growing bigger, wouldn't the ATP demand be higher? And the short answer to that is yes, it would. But some cells will grow bigger to try to provide a larger target for a cell signal, right? More receptors. We see that, for example, in the thyroid gland. Some of you might be familiar of the term a goiter. A goiter is a swollen thyroid. It's a hypertrophic thyroid gland. And it's growing bigger because it's not receiving the molecule iodine that it requires. So it gets bigger, trying to create a bigger target for circulating iodine. And on a temporary basis, that might work. Another cell adaptation that we see is hyperplasia. So hypertrophy is looking at a single cell and seeing that it's getting bigger. Hyperplasia is not only addressing possibly an increase in size, but also an increase in numbers of cells. So for example, red blood cells do this in altitude. If you go up to Denver or Mexico City, those are communities that are very high altitude areas. Your red blood cells, your existing red blood cells, don't carry enough oxygen because they don't get enough from the atmosphere. The lungs don't breathe in enough oxygen from altitude. So the answer is for oxygen delivery is to make more red blood cells. Each individual cell carries less oxygen, but now we have more of them. Does that make sense?
Yeah.
Okay. Now you can also have an adaptation that's called metaplasia. Metaplasia occurs when you get a change in cell type. We talked about this when we just mentioned the change of, when we talked about a callus. That's an example of metaplasia. So you're getting the cell type is going to, well, actually, that's a type of hypertrophy and hyperplasia. So metaplasia is the cell type is actually going to change. Meta means change. So the cell type changes. So for example, in our airways, if you are a smoker or you know somebody who smokes, their airway is going to change in response to that stressor, which is smoke and all the junk that's in that smoke. So normally, it's a simple columnar epithelium. It's actually, you guys, you probably, it's more correctly called a pseudo stratified columnar epithelium. But with smokers, it becomes a stratified squamous. So that's metaplasia. It changed from being columnar to squamous, from being a pseudostratified, which really means it's simple, to a stratified, because the stressor, smoke, caused an adaptation to occur. Now, it also means that this stratified squamous epithelium isn't going to be able to do what the columnar epithelium was designed to do. So this isn't good, right? It's not doing what it's designed to do, but it is resisting the chronic stress, this example being smoke. Now, we can also get an adaptation that's called dysplasia. So DYS tells you it's dysfunctional. It's not good. We don't want to see that prefix in front of words because it's indicating probably that there's something that we would rather not see happen. So what we're seeing in dysplasia is we're seeing abnormal size and shape of cells with lots of variation. So it doesn't show, when you look at nice squamous epithelium. You've got, if it's stratified, you got all the cells look about the same and they're layered up. It's pretty regular. In a dysplasia, you're going to see some abnormal shaped cells. Some of them are big, some of them are small. And this same tissue would now be described as being dysplastic or undergoing dysplasia because of the abnormal size, shape, and lots of variation in the cells. This is probably precancerous. With dysplastic cells, we describe them as being pre-neoplastic, because neoplasia refers to cancer cells. So cancer cells aren't going to follow any of the rules of any other cells. They don't listen to signals that say die. They don't listen to signals that say stop growing so fast. That's basically the definition of a cancer, is that the cells stop behaving the way they're designed to behave and they start growing out of control. So these are ways that cells can adapt. If they can't outlast and they can't adapt, then they're going to undergo death. And death in cells occurs two ways: through necrosis or through a term that's called apoptosis. Necrosis is messy. Apoptosis is clean, sort of organized. Some websites will describe necrosis as like cell murder, and apoptosis is cell suicide, except that it's pre-planned suicide. It's organized, it's for a purpose. It's part of homeostasis. We have a total number of cells in the body. Our heart stays about the same size. Our liver stays about the same size, because as cells undergo mitosis, other cells undergo apoptosis. So it's a way to regulate cell numbers. And it happens on a daily basis to the tune of tens of millions of cells every day undergo apoptosis. We'll talk a little bit more about that later on. Right now, we're going to talk about necrosis. Now, it's messy. It's often due to a stressor as opposed to apoptosis. And the primary stressor, the most common stressor for cells, is ischemia. Now, ischemia means decreased oxygen delivery to that cell, or more often, a group of cells. And so it's a fancy word that tells you that cell's not getting enough oxygen. We already know the pathway, right? We already talked about that pathway that occurs when there's not enough oxygen, because it's the same pathway that we described here. If there's not enough oxygen, the mitochondria is not going to be able to perform and produce enough ATP. We're going to get sodium-potassium pump failure. We're going to get hydropic swelling. So we already know the mechanism when there's ischemia as the stressor. So when ischemia is a stressor, we got decreased oxygen. If we have decreased oxygen, we know we're going to have decreased ATP. If we have decreased ATP, we know we're going to have a decrease in the sodium, potassium pump. And that's going to give us hydropic swelling. And if we get hydropic swelling allowed to progress, then that's going to give us destruction of the internal structures of that cell. We're going to get, the nucleus is going to disintegrate. It's going to release its contents into the cytoplasm of the cell. So we're going to have abnormal nucleotides and proteins inside the cytoplasm of the cell. We get breakdown of other structures within that cell. The membrane is going to break down and the contents of the cell are going to be released to the outside world. The complete destruction of the cell. So it becomes extra. So all of this is abnormal. And all of this means that what's going to happen is we're going to get inflammation from these damaged cells, and that's going to give us an immune response. We're going to get activation of the immune system. So that's where it gets messy. And it really means that an isolated area that wasn't getting enough oxygen is going to give us a system-wide response. The inflammatory response is going to initiate an immune response, which we know generally is a systemic response. It's going to become a systemic response. So it's a real mess. The contents of the cell were extracellular. They're not supposed to be there. We're going to get inflammation, and then we're going to get activation of the immune system. Eventually, it's going to get cleaned up. But in the meantime, it causes the patient is going to feel sick. They're probably going to have a fever. There's going to be loss of function in the tissues that are involved there. So there's lots of things going on here that are going to cause pathology. So we get inflammation. The patient's not going to feel very good. So they have malaise. They don't know why. They may be in a relatively small area. So they don't necessarily stop functioning, but they're aware that things aren't normal. Probably have a fever because of the immune response. The heart rate is up because they're stressed and there needs to be a repair process. And the white blood cell count goes up because they've been activated in the immune response. So this necrosis is messy. And we have different categories of necrosis. There is what's called coagulative necrosis. And what happens in coagulative necrosis, this is typically what we would see as a result of hypoxia, sometimes described as being non-traumatic injury, hypoxia, as opposed to a crush or a break, those sorts of things, is that the proteins that have been released, they are coagulated. There's the term. So a mass is created, a mass of denatured protein from the dead cells. And over time, it will be dissolved. There'll be some enzyme action, there'll be macrophages that will work on this mass. But in the meantime, it's causing symptoms. It's a mass of protein that's not supposed to be there. So we're going to have this immune activity. You're not going to feel good for days to weeks in some cases. And that mass of protein, before it's completely degraded, may provide a nice little focus for an infection, a nice little target of nutrients for a virus or a bacteria that happens to be circulating nearby. So that's coagulative necrosis. There's something called liquefactive necrosis. Very similar, but the enzyme activity, the lysosomes, the enzymes within the cell are so aggressive, they actually take the denatured protein a little further and create liquid-centered abscesses or cysts. So it's aggressive. Denaturation of proteins that form liquid-based cysts, otherwise known as abscesses. So it's very similar in terms of the sequence of events, but the aggressive nature of the breakdown of the proteins creates liquid. And they're also going to be dealt with by macrophages and eventually disappear. But in the meantime, they're causing symptoms. Now, there's lots of other types of necrosis. There's fatty necrosis that occurs in adipose tissue. There's caseous necrosis, and caseous necrosis is a combination of the first two that we talked about, a combination of coagulative and liquefactive. And when you look at it under a microscope, it looks like it's sort of white, clumpy masses that somebody thought looked a little bit like cheese, and that's where they got the name caseous from, like casein, one of the proteins found in cheeses, I think, is the way that works as far as terminology goes. So it occurs because there's an incomplete breakdown of these proteins, leaving some debris in kind of a lumpy, granular appearance. So in certain types of cell death, that's what it's going to look like. And then we have the gangrenous necrosis. Gangrenous necrosis is a type of coagulant necrosis that we see more often in the lower limb or in the GI tract, places where there's low oxygen availability. So this is a type of anaerobic necrosis. It's going to happen in places where the availability of oxygen is really, really low. So it's, again, going to be most commonly due to ischemia. The dead tissue can be described as dry, that's a dry gangrene, or if it's more liquefactive, it's called a wet gangrene. And it can be dangerous because it's going to spread more aggressively in those low oxygen environments. So in a low oxygen environment, the cleanup is delayed, and as a result, the gangrene, the death of surrounding cells can become progressive. So it's often when you hear about someone who's got gangrene, they need to have a limb amputated because they're concerned that the spread will develop before they can get the necrosis under control. Okay, now the next topic that we're going to talk about is apoptosis. Remember, we talked about apoptosis as being a sort of pre-planned or programmed type of cell death. So sometimes it's described as being a fundamental biological process. It's local, it's neat and tidy, so it's all the things that necrosis is not. And it's really designed to help maintain So it's basically designed to help maintain the homeostasis of cell numbers. When we're in utero, when we're developing in utero, our hand starts off as being like a mitt, where you've got a thumb, but the 4 digits are fused to one another. And through apoptosis, the cells between the fingers die off and give us 4 independent digits. That's an example of apoptosis. If cells go wrong, through mitosis that is abnormal, mutations, then apoptosis is often going to be instigated. It's going to be initiated because the abnormal cells send signals, and the genes respond to those signals and produce a protein that is going to promote apoptosis. We're going to talk more about apoptosis starting next week. Okay, so I think you can get from today that some of us need to do a little more studying of normal, right? We should all know the organelles of a cell. We should all know about ATP.